Hello and welcome to Best Practices for Robust and Reproducible Neuroimaging Meta-Analyses. This is going to be presented as an OHBM educational course, July 2023 in Montreal, Canada. So let's look at the goals for today. First and foremost, we want you to learn the motivations for neuroimaging meta-analysis. So why are meta-analyses important? Why do we need to do them in the first place? We then want you to get familiarized with the landscape of tools and methods for meta-analyses. So there are currently many options, and these different options are important for different research questions. So we hope that by the end of today, you know that for your research question, what is the right tool for the job. We also want you to understand the common pitfalls in statistical inference and meta-analysis. So how to, critically evaluate the results of a meta-analysis and the assumptions going into a meta-analysis. And finally, and this is very important, we want you to get some hands-on experience actually performing a meta-analysis. And we have a focus on robust and reproducible practices and systematic reporting standards. One note is that in today's course, we'll be primarily looking at fMRI meta-analysis, but very similar approaches can be applied to BVM and PET, and there are also emerging techniques for other modalities. So first we're going to start with 4 lectures to set up the scene. So I will first talk about motivations for meta-analysis. Then Katie Bodenhorn will tell us about conventional versus automated meta-analysis. Kendra Odick will then tell us about the decision tree for meta-analyses. And JB Pauline will tell us about the pitfalls and reproducibility in meta-analysis. Then we will switch to practicum. So this is going to be the hands-on part of the course. And James Kent will set that off by talking about Prisma and how to annotate studies. And Yifan Yu will tell us about meta-analysis specification and execution. In between these two talks, we're going to have some hands-on sessions so you can actually try things out yourself. All right, so let's get started with an overview and motivation for neuroimaging meta-analysis. First, I think it's important to take a step back and ask, what is the goal of brain mapping? What are the kind of questions we want to answer in general? So here's 4 potential questions. We might want to map the regions associated with a particular mental state. So for example, if you're interested in pain, you might want to know which brain regions are active for pain, which brain regions signal pain. Then we could ask a reverse question. We could ask, for a particular region, what functions does that brain region support? So for example, what mental processes does DLPFC support? And then we might want to look at not just individual regions, but also networks. So for example, what are distinct subcomponents of the default network and what do they do? And then we can ask these type of questions in different populations. So typically we ask these questions in healthy individuals to establish a baseline to understanding of how the brain works, but we could also ask in specific populations. For example, in a developmental sample, we could ask, how does cognitive control develop in the brain as you age? Or in a specific sample of people with a disorder, we could ask a question. For example, do people with anxiety process fear differently? Do their brain regions activate differently when they experience fear compared to a healthy population? And the way we typically answer these questions is by going from concepts to brain maps. So we want to map cognitive concepts to different brain regions, right? So typically we start with cognitive concepts. So we have different theories about different concepts that we think are different processes that are involving different regions. So we have ideas of what these may be. For example, something broad like cognitive control, something more specific like conflict adaptation, or something completely different like pain, reward processing, or even something that's not necessarily a state, but something we're doing to participants, for example, nicotine administration. So these are all valid places to start. And we typically then have a specific task that we think measures that cognitive concept. So for example, in the case of conflict adaptation, Stroop and go-no-go tasks are both measuring conflict adaptation. So taking a look at just the Stroop task, we could use that as a measure of conflict adaptation in a sample of participants. So for example, 20 participants undergo fMRI and they see a congruent condition and an incongruent condition. And the difference between the incongruent and the congruent condition is going to reflect the process of conflict adaptation. So we might then, when we perform this contrast between these two conditions, this analysis that we're going to call, we might see the DLPFC is active. So then we could say DLPFC is associated with conflict adaptation. So this is all well and good, but what are some potential limitations of this approach? And one of them really has to do with power. So every time you run a study, you're sampling from a population, and therefore you have limited power in how much you can detect of the true effects. So one way to look at this is with a simulation. So here we're looking at simulated effects in a population. And so these would be like the ground truth if we could know exactly, for example, with cognitive control, conflict adaptation, what are the brain regions that are active for this process. We might see a variety of voxels that are activated here. Some are going to be activated very strongly, very strong association with this task. Some are going to be deactivated very strongly. But we're also going to see some voxels that are, moderately activated by this process. So, in white we have outlined the regions that have the strongest association, but as you can see, there's a wide variety. So what happens when we take this real effect and we sample it? Because again, a study is just a sample of these real effects. And so here we have sampled this real effect with 3 different sample sizes, 20 subjects, 40 subjects, and 100 subjects, and then across 3 different corrections for multiple comparisons. Because again, you typically in fMRI doing massive univariate tests across every voxel, so you need to correct for all the tests you're doing, and there's different strategies depending on various reasons, but some are more stringent than others. So if you look at the top left, that would be the most stringent condition. So that would be one in which you're applying a very strong correction for multiple comparisons, FDR 0.05, and you only have 20 subjects. And in that case, as you can see, you're going to see a very small amount of the real picture. And these are not necessarily false positives. These are potentially real effects, but you're only seeing a small amount of the true underlying signal. And what that means is that when you get your results from that 20 participant study, you might only see those 2 regions. One region is active, one's deactivated. And let's say this is conflict adaptation and that active region is DLPFC, then you might say DLPFC is uniquely associated with this. This is the region that's really responsible for conflict adaptation. But of course, when someone else runs the same study and they take a different sample of this population, they might see a different region and they may not see DLPFC. So then you're like, why are these 2 studies seeing different results? And this can be purely due to chance. This is just purely due to the fact that we have limited power to detect effects. And so this is a problem, obviously, because that means that any individual study can only tell us so much about the ground truth. And now the question is like, okay, that was a simulation, but in the real world, is this a problem? And the answer is yes. So basically, and here in this paper, Russ Poldrack and some colleagues looked at real sample sizes out in the wild. And what they found was that over time, sample sizes have increased. So, in 1995, there were only 10 people per study. And in 2015, we had around 30. Now, the question is this enough? Is this a big enough sample size? And the answer is generally no. So even with 30 subjects, we only have about 80% power to detect very large effects. And so relatively large effect size of 0.75 is what you can reliably detect with a sample of 30 subjects. And the next question is, what do real, effect sizes look like? So do we have very large effects in cognitive neuroscience, or do we have smaller effects? And they estimated real effect sizes using the Human Connectome Project, which is a very well-powered study, so we can actually look and get a better estimate of what the true effect sizes for different tasks are. And the strongest effect sizes here at the top for motor tasks, we see Cohen's d under 0.75. So that means that even for very strong effect sizes, we're still underpowered. And so that means that for tasks that are more nuanced and perhaps lower hanging fruit are gone, so you're going to go for something more nuanced, you're going to have an even smaller chance of detecting. So even for gambling, that's a Cohen's d of around 0.4. So that's not good. That means we're probably going to miss a lot of true effects. But this also unfortunately means not just that you're going to miss on true effects, but you might potentially also see false positives. And so in this influential study from Catherine Button, Power Failure, we see that low power also increases bias, and studies that have a low amount of power also have a relatively greater amount of bias. And this is due to the Winner's curves, basically. So because of chance, the effects that you were able to see above threshold, you are basically seeing an artificially inflated effect size. And this means that you might see a very strong effect in DLPFC, and you think, great, this is a Cohen's d of 1.5, so I'm very confident that DLPFC is the driver of cognitive control. But in fact, there are many other regions that have similar effect sizes, and you're overstating your effect size. So this is a problem because we might come away with too strong conclusions from a single study. So okay, this is a problem, and we've been talking about this for years. People have been, at OHBM and other conferences have been saying this is a problem. So have we gotten better? Are we in better standing? And unfortunately, it doesn't really look like it. So although sample sizes have increased over time, in a recent sample from done using PubGet by some colleagues, they found that even in 2022, the average sample size was still around 30, maybe a little bit more than 30. So that means we haven't really improved much on having 80% power for anything much smaller than the Cohen's d of 0.75. So that means for smaller effects, which are probably the norm in cognitive neuroscience, we really don't have enough power. So okay, we're in trouble. So what do we do? And as you can, the idea here is that we should be going towards a cumulative science. So if you search fMRI on PubMed, you'll see that even now, fMRI studies per year are at an all-time high. So we're still publishing many and many of these studies, and many of these are underpowered. And so meta-analysis is a tool that we can use to aggregate these results, to have a stronger finding, to have a more sufficiently powered and consensus estimate of the true effects. So now that I've hopefully convinced you that individual studies have limitations, how can we use these individual studies and the rich literature that we have accumulated to build a cumulative science? And so the main tool of choice is meta-analysis. Meta-analysis combines findings from individual studies to build consensus estimates of brain activity for a given set of analyses. So the way this typically works is you first build a set of studies, which here we're going to call a study set, and from those individual studies you're going to pull out individual estimates, which we will then combine to build a consensus estimate. So here's a very nice example of this. This was in 2008, a very influential meta-analysis of emotion. So here they combined 162 studies to build a consensus map of emotion. Now to you this may not seem particularly surprising, but this is in part because of these types of meta-analyses that have been conducted. So this was able to very comprehensively map all of the regions of the brain that are involved in emotion. And this is a way to form prior knowledge. So meta-analyses help us build this cumulative knowledge that we can use in future studies. And I don't mean this just in the sense that we can use this to form theories, although that's very important. But we can use these meta-analyses as prior knowledge in future studies to increase power. So the way it works is that if you know that you're interested in a particular process, you could use a previous meta-analysis as an a priori mask in order to reduce the number of comparisons. So let's take a look at a specific sample. Here is a map of significant pain responses. This is a very well-powered meta-analysis that was recently published. So if you were looking at a study in which you were going to induce pain, but you wanted to look across distinct populations. So for example, let's say you had the hypothesis that individuals with anxiety would show decreased activity in pain regions as a response to pain. In order to increase the power to detect more regions that are pain-related, you could restrict your analysis only to the regions shown in this meta-analysis. And by reducing the number of comparisons, you now can use a more liberal thresholding method. You have to correct for fewer comparisons, and therefore you have a more liberal threshold, and this in turn increases power. So this is a very powerful technique for using meta-analyses in future studies for increasing the sensitivity of a study. So let's talk about some specific techniques for meta-analyses. And so the most popular technique, and the one you'll learn about the most today, is called coordinate-based meta-analysis, or CBMA. So again, we first have our study set of studies that we want to combine, and from those individual studies, we're going to pull out peak statistical coordinates that are reported in tables. So thanks to the fact that in brain science and neuroimaging, we report activities in a standardized space, which are either MNI or Tallarag space, we can put them together on common maps. So as you can see, individual analyses, like in this case houses minus faces, will report a set of coordinates which represent the peak activity for different clusters, and we can input those into a map. Now what we're going to do, because we only have the peak coordinates, we want to reconstruct the original analyses maps. So we do this typically with a kernel. A kernel will basically fill in a region around the peak that represents the originally seen activity. Obviously, this is an imperfect reconstruction, but this works relatively well. So now that we have done this for every study that we want to combine, we can now average those maps. And so as you can see here next, we have the average map, and this represents basically a proportion of studies that activate each region. This depends on the specific method, of course. There's ALE, there's MKDA, and there's other methods you'll hear about later, but the general principle is more or less the same. You're combining these reconstructed maps into an average map, and then finally you're going to run some statistics. So here on the right we have the final result, which is a threshold of statistical map. So there's various methods for this, but generally speaking, what you're trying to find is the regions that consistently above chance activate for this set of studies. So that is coordinate-based meta-analysis. And again, one of the key principles of coordinate-based meta-analyses is that you're taking these peak coordinates, so they are the peak of individual clusters that are reported in papers, and you're trying to reconstruct the original image. And again, this is because we don't actually most of the time have access to these original statistical images. So here's an illustration of that. We have 4 studies, and in those 4 studies we take the peak and we form a kernel around that peak. And then we combine those 4 studies and average them out. And in this study, they looked at to see how well did this coordinate-based meta-analysis approach approximate the true signal. So if we have 4 studies sampling from this true signal, and then we reconstruct the original images and average them to look at how closely does this map onto the original signal. And as you can see, it does a reasonably good job. So this approach, if you have enough data, will get you pretty close to the original maps. However, it's not perfect. Ideally, we would like to just have the original images. That data is more rich. So what if instead of having these reconstructed maps, we could have the original statistical images? And that is essentially what an image-based meta-analysis is. And image-based meta-analysis follow the same principle as coordinate-based meta-analyses, but again, instead of using these reconstructed maps using the peak coordinates, we get the original statistical images, and these can be either effect maps or statistical maps, so Z maps or T maps, and depending on the specific method that you use, the specific algorithm, you can combine these maps and do a image-based meta-analysis. And as a result, image-based meta-analysis has more power and more fidelity than CBMA. So as you can see here from this paper, they compared CBMA using various techniques and IBMA using various techniques, and the main takeaway is that with image-based meta-analysis, you get a much richer and more full picture of what is happening on average, and you also get more power. So basically you need fewer studies to find a consensus. Again, here's the combination of the different maps. So I guess the question is, why did I tell you that we're mostly gonna focus on CBMA and not IBMA? And the reason is that unfortunately, although there are places to deposit your statistical maps, which you might have heard about, for example, NeuroVault is a repository of statistical maps for published papers, the vast majority of papers still only report coordinates. Only a small proportion of studies actually deposits their original statistical maps onto a repository where researchers can access them. So as a result, right now, I estimate that we have around 1,000 to 1,200 unique papers with statistical maps shared, whereas we can get access to around 20,000 or more papers that have coordinate tables. So because of this, we're going to focus primarily on CBMA, but I did want to let you know about this because it's important for you to share statistical maps if you're involved in running a study. This really helps with meta-analysis. And it's really important to also add informative metadata so people in the future know which maps were important ones, which were not, which were the contrasts of interest, what are the different things you were testing, makes it easy for meta-analyses to use your data. But also to let you know that this is why we primarily still focus on CBMA, but in the near future there should be enough data to do some more image-based meta-analysis. Okay, so now let's take a step back. I just told you about IBMA, CBMA, and what are the different overall techniques for doing meta-analysis. But the good news is, for the most part, those techniques have been, well researched, and there's, good tools for applying them. So you, as someone who's performing a meta-analysis, the main thing you want to worry about is what actually goes into your meta-analysis. So a very important part of a meta-analysis is forming a study set, or forming a collection of studies or publications that you want to meta-analyze. And this process is critical because the assumption that goes into a meta-analysis is that all of the inputs to a meta-analysis represent a common process or a common mental state. You're basically saying all of these studies have something in common. And so it's very important to be systematic and to be transparent about what criteria you used to include studies into meta-analysis. This is such an important problem that there is a way to report this called PRISMA, which is the Preferred Reporting Items for Systematic Reviews and Meta-Analyses, which is basically a methodology for specifically excluding and including studies at different stages based on different reasons and coming out with your final study set. And so I really encourage you to read this paper, Mueller et al., which is called 10 Simple Rules for Neuroimaging Meta-Analysis. It outlines these guidelines and more in very clear detail, but I wanted to point out two specific things that I think are really important. When you're doing a meta-analysis, you need to be specific and transparent. So you need to think about what am I actually trying to meta-analyze and make sure you report that clearly. So for example, am I trying to do a meta-analysis of all executive function measures? So you could collapse across MBAC tasks, Stroop task, go, no-go task. Or am I only wanting to look at Stroop? For example, you might only include studies that looked at specifically the Stroop task. And the results of these two meta-analyses are going to mean different things. The one that includes all executive function measures might only look at regions that activate for EF more generally, whereas the Stroop task is giving you a more specific result. So keep that in mind. But also keep in mind that generally you have a trade-off between power and specificity. So if you choose to only look at Stroop tasks, you have a more specific meta-analysis, but you probably have fewer studies to choose from. And this is important because, this is just a general rule of thumb, but you generally, for a CVMA, you want to have around 70 to 20 experiments to avoid a small number of studies driving effect. Again, this depends on the technique, etc., but this is a good rule of thumb to keep in mind. And again, finally, be systematic. So think about these questions advanced before you do a meta-analysis, write them down, come up with a plan for how you're going to select studies, and make sure to keep track of all of your metadata so you can report this clearly. And later on, we're going to talk about some tools that make this easier. So in summary, individual studies, especially in fMRI, are significantly underpowered, and it remains so. So this gives a chance of reporting false negatives as well as false positives and inflated effect sizes. Meta-analysis is a readily available tool for synthesizing the literature, and it can help with the problems and limitations of individual studies. Coordinate-based meta-analysis is the most common method due to data availability, but also has limitations. It has lower power, and it doesn't give you as rich of a picture of the original brain activity. Image-based meta-analysis is superior, but so far has been unpopular due to a lack of data that we have access to, although this is soon to change. And finally, one of the most critical scientific aspects of a meta-analysis is the inclusion criteria. So think carefully about your research question, be systematic, and report transparently. And we're going to focus a lot on this very last part because it's, I think, one of the most important parts. So thank you for your attention. That concludes the introduction to best practice for robust and reproducible neuroimaging meta-analysis. Next up, Katie Bodenholm will be telling us about conventional and automated approaches to neuroimaging meta-analysis.